So, my name is Adrian Stanley. The title of my paper is Cognitive Enhancement, to be or not to be. So it's about the role of the NMDA receptor subunit NR2B in cognitive enhancement. So, the first thing I'm going to start talking about is the learning process, how learning occurs in the brain. So that's going to involve a process called LTP and a process called LTD, which I'll go into more detail about later. And of course, the main area where short-term memory is converted into long-term memories is the hippocampus. So I'll talk about the hippocampus and its anatomical structure. And I'm going to talk about the glutamate receptors AMPA and AMPA. And then I'm going to talk about the subunits. And I'm going to talk about what happens when the NR2B subunit of NMDA is overexpressed, and then I'm going to go into future directions. Alright, so learning, basically whenever you learn things, it's like the main thing that's happening is an increase or a decrease in the synaptic efficacy. So basically every time you're told something, your cells the cells associated with the thing you're told become more easily depolarized, and that's basically what learning is about. So the process in which it becomes more easily depolarized is called LTP, and the one where, in an instance where it becomes less easily depolarized, is called LTD. So, and of course, one of the main areas that it occurs in is in the hippocampus. So in the hippocampus you have, this is a cross-section of a rat hippocampus. So this is the entorhinal cortex here. It has axonal projections that project into the dentate gyrus of the hippocampus. And the dentate gyrus of the hippocampus has axonal projections that project into the CA3 region of the Amen horn. This piece is the Amen horn of the hippocampus. And the CA3 has projections that project into the neocortex, where memories can be stored for long term, and they have axonal projections that project to the CA1 region over here. The CA3 to CA1 axonal projections are called the Schaefer collateral pathway, and that's going to be important later when I discuss how they cut the hippocampus out to do their LTP tests. Alright, so in long-term potentiation, the increase of synaptic efficacy, it can happen in a number of ways. It can happen at glutamatergic synapses, which I'm going to discuss today. That's the primary way it happens, because glutamate, of course, is the most abundant neurotransmitter in the brain. It can happen to G protein coupled receptors, can activate secondary messengers that increase synaptic efficacy. You can have retrograde neurotransmitters like cannabinoids and nitric oxide that can increase synaptic efficacy, but this is a presynaptic efficacy. And also you can affect efficacy by affecting the voltage gain and calcium channels, but of course I'm talking about this one. Alright, so at glutamatergic synapses, you have the AMPA glutamate receptor and the NMDA glutamate receptor, AMPA. So basically, when the presynaptic cell releases glutamate binds to both AMPA and NMDA. But once it binds to AMPA, it opens the channel and allows an influx of sodium ion, which depolarizes the cell. When glutamate binds to NMDA receptors, basically initially the NMDA receptor doesn't open. It has this magnesium ion blocking the channel. But as the intracellular space becomes more depolarized from the activation of AMPA receptors, then since the inside is more positive, it'll repel the magnesium out of the channel, and as a result, that channel opens. But this channel, different from AMPA, in addition to allowing sodium in, it also allows calcium in. And calcium is an important secondary messenger in the LTP process. So as you can see here, after the NMDA channel opens, then calcium will bind to this protein called calmodulin, and then calmodulin will bind to this kinase called CAMK2. And then CAMK2 can either phosphorylate AMPA receptors so that whenever glutamate binds to them, they'll stay open for longer, and they're more reactive. And calmodulin can also it can also phosphorylate itself, so that it'll remain active even after the calcium concentrations are lowered. And if it does that, then it can keep phosphorylating AMPA receptors even after the NMDA channel closes. Calcium can also activate protein kinase C, And protein kinase T can go down to activate, induce the transcription of certain genes, and those genes can result in the production of proteins that leads to the formation of entirely new synapses. So that's an even more longer term increase in efficacy between the two neurons. So this is calmodulin, the CAMK2, calmodulin kinase 2 protein. So basically, the calcium will bind to calmodulin, and the calmodulin will bind to the kinase, and now the kinase this kinase right here can go on and activate things, but if it gets phosphorylated, if it phosphorylates itself, then it can remain active until eventually, of course, the phosphorylation wears off and goes back to normal. So yeah. So PKC, it goes on to activate CREB. So CREB2, it blocks gene transcription. So whenever it binds to CREB regulating, it binds to this gene right here, and it prevents transcription of this gene down here. So whenever you have CREB1 phosphorylated and bound to the CREB response element, then it'll lead to transcription of the gene. So that's what PKC does when it gets activated. So LTD, long-term depression, is basically the opposite process. So basically, the synaptic efficacy is decreasing. So whenever it's responding to the same stimulus, it'll depolarize even less. So what determines whether it's going to undergo LTP or LTD is the frequency of stimulation. So if this postsynaptic cell is constantly stimulated and there's lots of calcium, there's high calcium concentrations, then it'll lead to activation of the protein kinases, and that'll lead to production of LTP. Whenever it's low frequency stimulation, you have low concentrations of calcium, and instead of calcium activating calmodulin and PKC, it'll instead activate phosphatases, which can actually removes phosphates from the AMPA receptor to make it less responsive, and can even lead to the downregulation of the AMPA receptors on the membrane, which decreases the efficacy. Alright, so the NMDA receptor subunits. Every NMDA receptor has a NR1 unit, and it has a variable NR2 unit. The NR2 unit can either be 2A, 2B, 2C, or 2D. The receptor subunit 2C and 2D, they have low magnesium dependence, so they're less likely to be involved in the LTP process. But these ones have high magnesium dependence, so these ones are the main ones that are associated with LTP. Yeah. So NR2A versus NR2B. NR2A and NMDA receptors, they open faster, but in addition to opening faster, they also close faster. So that it initiates LTP to a lesser extent because, of course, if it closes fast, then there's only going to be a little bit of calcium entering, so it's less likely to induce LTP. Basically, when you're young, you have a higher concentration of NR2A, or when you're young, you have a low concentration of NR2A receptors, and as you age, you get more and more of them. For NR2B, they open slower, but when they're open, they're open for a lot longer. So it induces LTP even better because there's a lot more calcium in the intracellular space. So this is the opposite of this. So the concentrations of this is high when you're young, and it decreases as you age. So there could be some kind of implication there about how, like, the way children learn relative to adults. Alright, so basically in this paper, they did these tests right here. They created transgenic rats that overexpressed the NR2B gene, and then they put them through a few tests. They put them through they tested their hippocampus to test the LTP at the Schaefer collateral pathway, and they gave them the novel object recognition test to test their recognition memory, and they put them in a Morse water maze to test their spatial memory, and a teammate to test their working memory. I'll describe those tests right now. Alright, so in the hippocampal recording techniques, basically, the way the hippocampus is arranged, you can basically, it's arranged in such a way that you can slice it out of the brain, and all the axonal projections that I described to you earlier will still be intact. So what they do is they take an electrode and they put it in the CA1, they put an electrode in the CA3 neurons, projecting to the CA1. So they stimulate those neurons, and they measure the depolarization of the postsynaptic cell, and they see whether or not the efficacy is increased or not. So here in these two graphs, they're testing the presynaptic responses. So this is just the response of the presynaptic cell to the stimulus. So as you can see, they're both the same, so there's no change in the presynaptic stimulation. But in when they look at the you look at the postsynaptic cell, then you can see that after they give the high frequency stimulation of 100 hertz, the response of both of them, both of them undergo LTP, but in the transgenic rats, they undergo LTP to a greater extent. So they maintain a higher depolarization in response to the same presynaptic stimulus as opposed to the wild type. And when they introduce the low frequency stimulation, they see that there was no real change. So long-term depression wasn't really affected in the transgenic rats and overexpression of the NR2B. So that suggests that NR2B is critical for LTP. So here they did the same thing, except they introduced antagonists to the receptors, so they can make sure of the importance of NR2A and NR2B in inducing LTP. So they use this drug called NBP-AAM0777 to antagonize the NR2A subunit, and they use the RO256981 molecule to inhibit the NR2B subunit. So what they notice is when they introduce for the wild type, they introduce the NMDA, the NR2A antagonist to the wild type. They noticed there was a decrease in LTP, but they still maintain some LTP. For the transgenic, there was also still a great reduction in LTP as compared to no drug, but it was still significantly higher than for the wild types. So yeah, you can see the difference in EPSP in transgenic versus wild type for the drug and no drug. And for the NR2A antagonist, NR2B antagonist, they noticed that same thing happened. There was a decrease in LTP, but to an even greater extent here for the transgenic rats, because of course the NR2B was overexpressed with them, and you can see the comparison here. Then they introduced both of them. So by introducing both antagonists, they noticed that for both the wild type and the transgenic, LTP was pretty much completely stopped. So they're responding the same way as they did initially to the stimulus here. So LTP was completely stopped when both the antagonists are put in. So that means that indicates that NR2A and NR2B are critical for LTP, especially at this part in the hippocampus. Alright, so in the T-maze test, basically what they did is they have this maze shaped like a T, and they gave the mats 2 runs. The first run is a forced run, and the second one is a choice run. In the first run, they basically block off one of the wings of the T-maze, so they have to go down the other wing. So as a result, when they go down the other wing, they give the rat a reward. So they give it some food. And on the second run, they put the rat in the maze again, but this time with both of them unblocked. If the rat goes down the direction opposite the way it went last time, then they give it a reward. So they did that with these rats, and they made sure that they got their performance over 85%. So that 85% of the time they're choosing the opposite wing, and then they put a delay between the choice run and the first run, the forced run. So during the test they did a 50-second delay, but they tested it with a 1-minute delay, a 3-minute delay, and a 5-minute delay. So in the 1-minute delay, they are pretty similar in their ability to remember which way they went last time in order to go the opposite. In the 3-minute though, there was a significant difference between the transgenic rats and the wild-type rats. So the NR2B is allowing them to remember for even longer where they went last time in order to choose a different path. So this is really testing their working memory, their working memory. And at 5 minutes, it sort of like levels out again. They kind of like have the same response. In the novel object recognition test, basically what they do for this is they introduce the rats to, for the training session, they introduce the rats to two objects they've never been introduced to before, and they measure the amount of time they spend exploring each object, and then they divide by the total time they spend exploring both objects. So exploratory preference of about 50% means they explored both objects equal amounts. So as you can see during the training session, they both explore both objects in the same amount. So that says that it's not like the transgenic rats are more curious, so they're exploring more and they're learning more. It's just they're just as curious, they're exploring just as much. And over here, as you can see, when they introduced the next day, when they gave them one of the objects that they recognized and a new object, basically they measure the amount of time they spend exploring the new object and divide by the total time they explore both objects. The second object is an object they've seen before. So after an hour, both of them pretty much recognize the old object was something they've seen before, and they spend a lot of time exploring a new object. After a day, the transgenic rats were to retain memory of the familiar object for longer, because they explored the familiar object less than the wild type did. And of course, at 3 days, it was really significant because the wild type pretty much forgot about it, and they're exploring both objects evenly. And the transgenic rat still pretty much remembers it. And at 7 days, it equals out again. So they both recognize the old object and the new object as new objects. All right. So in the Morse water maze, basically what they do is they put these rats in like the circular pool, and the pool is like opaque, so they can't really see. And the pool is divided into 4 quadrants. And one of the quadrants is a platform that's about 2 centimeters, about below the surface. So they put the rats in the water maze. They let them find the platform, and then they put them in again and see how long it takes for them to remember where the platform was and go back to the platform. So in the training sessions, they measured the amount of time that it took for them to find the platform, and they noticed that for the transgenic rat, they spent less time trying to find the platform, especially around the second and third training sessions. They did around the third day. What they did was they had cues around the Morse water maze, so that the rats can identify them, and when they put them back in the maze, they can identify certain parts to see if they can figure out where the maze was before, see if they remember like the cues. And they noticed that both the transgenic and the wild type, both pretty much when there's full cue conditions, they both spend most time in the targeted quadrant. But when there's only partial cues, only some of the cues were there. The transgenic rats spent significantly more time in the maze in the quadrant that contained the platform than the wild type. So in conclusion, LTP and LTB are processes vital to learning. Learning primarily takes place in the hippocampus. NR2B is more efficacious in inducing LTP than NR2A. Both NR2A and NR2B don't show significant differences in LTP induction. NR2B overexpression enhances spatial reference, spatial working, and recognition memory in rats. And the NR2B gene, as a result of this, is a good target for cognition enhancing drugs. I know that they've been doing research into, like, they noticed that increased dietary magnesium tends to increase genetic transcription of the NR2B gene. They also noticed that they did, like, because the NMDA receptor is constantly being degraded and replaced again. So what they can do is they can either, like, inhibit the degradation protein that degrades it, so that as a result there'll be more NMDA receptors when new ones come on, or they can increase transcription of the transport protein that transports the NMDA receptors to the membrane. And yeah, that's it.